You know, son, I told you, tell me the death of my grade.
We're not doing.
Oh, okay.
Area.
Hold on. 9. $25 and 10. Okay. Yeah. predicting what days are. And I'm like, I need to look at a calendar for next week, I think. 6, yes. Right. Just to go over really quick while everybody's getting settled and signed in on the Kahoot. Just a reminder, today is Monday, 9/29, and lecture, we're doing chapters 9, 25, and 10. We're gonna try to spend most of our time on 10. So we're not gonna complete the whole Kahoot for chapters 9 and 23. Both 9 and 23, I have 10 question Kahoots. We're gonna go through a few of those questions. We'll probably knock out 10 questions altogether for 9 and 25, and then try to focus the rest of the time on 10, because obviously 10 is the meatiest of the chapters, especially today. 6 and 10, like I said, now we're seeing it. Now that we've done our concept maps for 10, we're seeing why. So that's going to be that. Again, so one thing that we'll talk about it in a little bit. I don't want to get ahead of myself. We'll talk about what is expected of you for chapter 10 when we talk about aerobic respiration, when we talk about glycolysis and Krebs, what I'm expecting you to know from those, which is not every step in it, not even close to that. But if you're not getting the major ideas of what the point is of those, and what the overall, like what we start with and what the outcome is of each of them and why each of them happen, that's the goal of what I want to take home from chapter 10 with aerobic respiration. Obviously, we've got enzymes and stuff like that to go over too. But anyways, the lab, we are doing lab 10. We're going to be hard pressed to take up the whole lab time today. So it's one of those days where we are probably, so it's a lot of concept in lab 10. So we're getting our last results from our Kirby Bauer, and we are going to be talking about the DNA stuff in the lab. We don't have the time to actually do any of the DNA experiments because they all take days or hours and hours and stuff like that. So we're just going to talk about the concepts of them, and you'll be tested on the concepts of them on the lab exam, okay? So we will go through it. There's a worksheet that we'll do. There's like a summary of DNA stuff in the lab, and I'll walk you through what I'm expecting you guys to know from that. So there's a lot of concept in lab and not a lot of doing in lab today. So because of that, in lab, we're going to walk through that Kahoot like we usually do in lecture. I'm not going to call on you for it, but I'm going to expect that we're going to be kind of participating and discussing it a little bit, just because it's a lot of concept kind of all at once. It all relates, a lot of it, to chapter 6 and 8. So hopefully it's not new stuff. But anyways, that's what we'll be doing in lab 10. Then on Wednesday of this week, obviously we don't have lecture on Wednesdays. Wednesday of this week, we will be doing lab 11, which is the ELISA, which is also a lot of new concept. It's going to be talking about like antibodies, antibody-antigen interaction. And if you don't know what antibodies or antigens are yet, that's okay. We're going to talk about it when we get to lab 11. Okay. All right. So then on Monday next week, we will do in lecture, we will be doing chapters 13 and 14. Chapters 13 talks about infection and disease and a lot of like terminology associated with that. What's the difference between a sign and a symptom? And there is a difference between those terms, and we absolutely need to know how to apply those kinds of terms, as well as like the infection process and you know, how, you know, organisms and pathogens get into our body and then multiply and then get out of the body and all that. So infection and disease is chapter 13. Chapter 14 is epidemiology, so a lot of new terminology for that one as well. And then in the lab on Monday, 10/6, we're going to be doing a review. This is a required attendance day, but you can do it via Zoom if you want. So on Monday, 10/6, you may want to join via Zoom on both of these if you want to. As long as you attend, I don't care, you know, this doesn't mean watch the recording. That doesn't count as attending. If you attend live in person or via Zoom, I'll let you decide how you want to do that, okay? But both of these can be attended via Zoom, and I'll send out an announcement about that towards the end of this week, just to remind you guys about that for next week, okay? But then Wednesday, 10/8, obviously we don't have lectures on Wednesdays, but we do have our lab midterm.
So on Monday, we have in class too.
This Monday,
Yeah.
yes, so there's lecture that
Okay,
day.
okay.
Yes, there's lecture that Monday, yes.
Do you have a study guide based for the lab intern?
Yes, so it is in the Canvas page. Let me pull it up real quick. This is in the way. Oh, no. I'll show you guys just so you all know where it is. So that there's no confusion about it. $844. Whatever. So you can find them in the modules. They'll be at the bottom of all of the modules, all of the weekly modules. Just like we have our lecture study stuff at the bottom, it'll be at the bottom of those. So this is week 7. So you can find it there, but you can find it at the bottom. So I have week 6 open, for example. Let's just scroll to bottom of week 6. Lab midterm study guide. There's 2 different ones. One of them is more like question based, and the other one is like draw a concept map or something like that. It's more like interactive based. And so you can do both or you can do either. I don't really care, but it's the same idea. So those are study guides for that. So proper study guides for those ones. And then we do have learning objectives for the lab that you can, you know, base your studying off of as well. Okay. Any questions? And there's study sheets too. Again, on every week that'll be available.
Okay, perfect.
And then this is the same exact study sheets that are in our lab manuals. It's just only the study sheets instead of interspersed with the labs, you know? Anyway, so that's all there too. So good study resources for that. Any questions about that? So we do have lecture on Monday, right? And we'll have a review for that on Monday. You can Zoom both of them if you want to, I don't care. But I will be moving to the lab. So they'll be, you know, it'll be on the same Zoom platform. It's just I'm moving my body to a different location in case you show up in person in the lab. All right. So it's up to you how you want to attend. I don't care. Then on Monday, 10/13, we will have for lecture, we have the Unit 2 exam. This marker just already gave up on me. Unit 2. And we won't have lab on that Monday. So Monday is 10/13, Tuesday is 10/14, Wednesday is 10/15. There is a lab on 10/15. Okay, so I know that 16 and 17, 10/16 and 10/17 are, is there a lab on 10/13? Now you guys, now I'm not sure. There's not, okay. I'm not sure how I scheduled it.
All break the end of our week.
Yeah, and so I think I scheduled us to have that Wednesday off
You did.
to match it up. Yeah. I think you're right. Just to be sure, I'm going to scroll down to the schedule. Yep, okay. 10/13. Okay, we do have lab that day. So, yeah, we do have lab on 10/15. So you do know that 10/15 is not fall break. Okay, so I have scheduled the lab that day. At the same time, let's not do lab on 10/15. I'm like looking at it and I'm like, we can fit in what we're doing on 10/15 on 10/20. So I'm gonna give you guys off on 10/15, okay? Wednesday, 10/15, that's another day of your fall break for my class anyways. Nothing for 10/15, so I'll probably have to send an announcement about that too. Just doesn't make sense. Okay, so anyways, this is what we're looking at, just the FYI so that we're clear. This is this week, next week, the week after. Okay, I'll give you guys Wednesday off on 10/13. 10/15, excuse me, 10/15. Obviously be here on 10/13. That's the exam. Any questions about the schedule? So just so that we know what's coming up and what to expect with that. Okay, so we know where our study stuff is on here. All right, make sure we're signed in onto the Kahoot. We're gonna get going with chapter 9. So we're gonna try to keep 9 and 25 a little bit brief. We can spend at least a full hour on 10. All right, let's see. 1, 2, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 16. All right, we've got people online too. All right, that's everybody. So we're gonna go ahead and get going. Is Jimena here today? Okay, you'll be our first one. Okay, the acronym CHOMPS. CHOMPS is used to refer to the elements needed for life. What is S? All right, so it's sulfur. So we have carbon, hydrogen, oxygen, and Jimena, what's the N in CHOMPS? Not, yeah, I don't know if that was you that said that, but okay. Did you hear what it was? Do you know what the N is? N is nitrogen. Okay, and probably, so this is one that we're gonna get at here in a second, with the nitrogen cycle, which I know some people were like, oh my gosh, with the nitrogen cycle, like, you know, what do I need to know about ammonification and nitrification and denitrification? Not as much as you think. I definitely don't need you to memorize those pathways, like down to like which atom is going where, and then I don't need you to even know the exact, like, what I'm trying to say, chemical equations for them. I know that's a chapter 25 thing, but I do want to touch really quick. Now we're on CHOMPS here. So obviously we know carbon and hydrogen and oxygen seem like obvious ones to us, right? Especially whenever we're thinking of organic molecules. But if you're leaving out nitrogen and you think it's some small little part of you, you will literally die without nitrogen. You can't without nitrogen, right? And so okay, so I'll say this. Anybody can name one of the macromolecules that has to have nitrogen in it? Maddie? Nucleic acids. And so specifically, when we're thinking of nucleic acids, we're thinking of the nitrogenous base for DNA and RNA, right? And ATP. When we think about ATP being our energy source for the cell, it is literally where the source of energy comes from. ATP is adenosine triphosphate. That means it's an adenine with a sugar. That makes adenosine by itself. And then the triphosphate's there. So obviously we need phosphate, which is the P in the CHOMPS, right? And then nitrogen is for those nitrogenous bases there. All right. So we'll get you again, Jimena, on a different question. So we did that. Okay, I'll just ask you the P then, because we didn't get to, I hinted at it. Do you know what the P is going to be, Jimena? There we go, phosphorus. So specifically phosphorus here, we're talking about the element phosphorus, but we find phosphorus in phosphate, obviously, which is going to be in our DNA and our ATP and our phospholipids, obviously. The other thing that we need nitrogen for, to come back to nitrogen, we talked about it being in our DNA, in our nitrogen spaces, but don't forget your proteins, right? Your amino acids. Amino literally means NH2 group, and amine group is an NH2 group. So you can't have DNA, RNA, ATP, or protein without nitrogen. So let's not forget the importance of nitrogen. And I guess briefly, while we're talking about it, I'll go over the nitrogen cycle just because I'm already blabbing about it. All right. In the atmosphere, what kind of nitrogen do we have? N2. It's N2, right? Just proper nitrogen gas, right? In the atmosphere. All right. N2 can be turned into NH4+, also known as ammonia, by bacteria. All right. Anybody know the name of that process? when we're turning into, okay, so ammonification. So what's your name? Okay. In there. So I don't call on you again. Here you are, Lippy, right? All right, good. So that's ammonification. That's all you need to know about ammonification. All right, so bacteria do this, all right? And also in whenever we are eating food, we can poop and pee out ammonia as a waste product as well from breaking down other nitrogen sources, like organic nitrogen sources that can lead to ammonia. That's not ammonification, but it can make ammonia as well. Ammonia can then be turned into nitrite, which is NO2, and then nitrate, which is NO3. And then this whole entire process. Anybody know what that process is called? Okay, beautiful. I saw you say it. Okay, Jordan, okay. Nitrification. Beautiful. And then plants can use this as a source of nitrogen. And then we eat plants or animals that ate plants. And then we get their organic molecules like DNA and RNA and protein and ATP whenever we eat them. Now it sounds weird, but this was the thing that I brought up in the later part of last week in at least one or two of my classes. We were talking about the GMOs and the transgenic like organisms in general. Okay, you guys, just to FYI, we eat DNA. Yes. Yeah. If you eat an animal or a plant, you eat DNA. Sounds weird, but you do, right? So that DNA, your body doesn't, incorporate normal chicken DNA into your body. Why would it incorporate transgenic chicken DNA into your body? It's still literally just DNA. So that was another thing that we went into. Like that's a thing that doesn't happen with GMOs or transgenic organisms. You don't incorporate their DNA into your DNA. That's completely ridiculous. So if that's why you're worried about a GMO or transgenic, that's not a reason to be worried about a GMO or a transgenic. What is a reason to be worried about a GMO or that's impossible, that's facts. That's not opinion, that's a fact, okay? What is a reason to be worried about GMO or transgenic is the fact that they are affecting, like, let's say plants. We have made transgenic corn, which I can guarantee you have not eaten non-transgenic corn, okay? I can guarantee you that all of the corn that you have eaten is transgenic. But much of the corn, probably 95% of all corn on the market now, is transgenic to contain a gene from bacteria that helps it resist basically insects from eating it. Okay, it's called, I think, BT corn, and it's Bacillus thuringiensis, and they took a gene from that bacteria and put it into the corn. It's transgenic now, and now bugs aren't going to eat the corn. It saves you from pesticides, so that they don't have to put as many pesticides on the corn. And you might think it's a good thing. Sounds good, yes? Except that plants can exchange their genes with other plants. Not with you, but with plants. And so that means that the corn that is transgenic can now give pest-resisting genes to weeds and trees and things like that. And now caterpillars and other insects can no longer eat those plants. And now we have screwed up the ecosystem royally. Yes.
So nitrogen fixation.
Mm-hmm.
Difference on that.
It's nitrogen fixation and nitrification are basically going to be the same thing, essentially. Okay, so nitrification and nitrogen fixation, essentially the same thing.
You're just breaking it down into a more simple.
Yeah.
From ammonification to nitrification. It's just a more simple thing.
Yes. And so then other bacteria and other, I would say other bacteria can convert nitrate into N2 back into the atmosphere. This was the last step. Anybody remember what that one is called? Denitrification, yes. Some plants can use ammonia directly, but this is very rare. Most of the time, if I say a plant can use ammonia, like we think about ammonia being in our fertilizer, yeah? And you're like, okay, yeah, there's ammonia in your fertilizer, so obviously the plants can use it. No, the bacteria that are incorporated in the plant's roots can use it and turn it into nitrate, and then the plant can use it. Plants use nitrates typically. So this is a little bit of not really a direct, honest way of thinking it. So I would leave it as how I drew it originally, just FYI. I would leave it as atmosphere becomes into NH4, which is ammonia. Anyways, you guys get this idea. This is literally the extent of what I need you to know about the nitrogen cycle. I don't need you to know all of like the equations down to the details or anything. Is this good enough for everybody? Okay. So since we were talking about chomps, I figured we would cover that. So I'm going to go ahead and erase this. Everybody good with that? Good, beautiful. Excellent. Right. I know I don't cover that very much in my lecture. All right, which of the following is heterotrophic? A heterotroph. When we start talking about the trophs, there's basically two kind of ways we can break down where organisms are getting their carbon or their energy. Carbon or energy. All right, your energy can come from the sun. And your carbon, you can make your own organic carbons from your, obviously sun, photo, sunlight. If you are making your own organic molecules of carbon from inorganic sources, so we agreed CO2 is inorganic. If you didn't know that, that shouldn't be news to you at this point. We learned that in chapter 2. Okay, so carbon dioxide is inorganic. If you're turning that into organic carbon, then we call you an autotroph. Excellent.
What makes CO2 inorganic? Is it just the fact that it's a gas?
So that is a much more complicated question than we ever teach you guys. What it has to do with is how electrons are being shared with carbon or how they're being hogged from carbon. And when we have hydrogen and carbon bound together, then we have an equal sharing of the electrons from hydrogen, which is very unique with carbon. And so that's what makes an organic molecule. And that's why we need the hydrogen to call it organic. So just having carbon is not enough to call it organic. We have to have that association, that equal association with the hydrogen. So it has to do with, and that's, again, me watering it down. But it has to do with how the electrons on carbon are being shared equally with something else. So that's
So there's
really,
not as many hydrogen bonds for an inorganic molecule versus an organic molecule?
well, whenever I were to, if I were to draw you a benzene ring, for example, right?
Mhm.
We'll just put the circle. You get the idea what's going on here. It's just carbons and hydrogens. No opportunity for hydrogen bonds. Not true hydrogen bonds, attractions between hydrogens and something else, because all of these hydrogen bonds are sharing equally with the carbon that they're attracted to. They're, I mean, literally covalently bonded to. So they're not being positively charged to create that. But the fact that they aren't being positively charged and they are just normal hydrogen having a normal covalent bond with something else, being a carbon specifically, that's what makes it organic. And so having the hydrogens that aren't being polarized by an electronegative molecule like oxygen is really what dictates an organic molecule there. So oxygen is electronegative. Now, if we were to look at how close carbon and oxygen are on the periodic table, pretty much the further right you go, the more electronegative you are on the periodic table. So fluorine is more electronegative than oxygen. Nitrogen is you know, the next one, and then carbon is the next one. So carbon does hog a little bit from some atoms, but as it turns out, the bond that it creates with hydrogen tends to be really stable and equal. So there's no polarity going on there. It's just how it interacts with the hydrogen, and that's what makes it organic. that specific interaction between those two atoms. It's more complicated than we need to get into. And it's obviously the reason why you guys didn't talk about it in GOB, because it's like
That's like,
too much. It's more like a physics chemistry type of a thing. Yes. But yeah. So remember, organic, we need to have hydrogen in there. All right. So we got Deontay answered a question. I heard you. All
Thank you.
right. Trying to find you here. Whenever I had them randomized like this, I have to actually look for people's names. Okay, so this is the idea. If you make your own organic from an inorganic, what does that? Like, okay, so I'll ask a question about this one. Isabella, all right? So which of, like we say that if the heterotroph is the mouse, why are the other three not heterotrophs? What would they be? Autotrophs. And so that's easy enough, right? And so that's the difference here. Heterotroph and photo and chemo and all of this stuff having to deal with where we are getting our carbons and where we are getting our energy from. So energy, if we're breaking down chemicals, which we talk more about that in chapter 10, right? We talk about breaking down molecules to release energy. There we go, yes. So we talk about catabolic and we talk about anabolic reactions, right? So if you're putting together molecules and you need energy for building things, then that makes sense. If you just break it down that simply, if you're building something, you need energy to do that, right? But when we break things apart, it releases energy. And so that's what chemotrophs do. They break chemical bonds apart to make energy. That's what we do. So we're chemotrophs. A heterotroph gets their carbon from organic sources that were made by autotrophs. So that's where we get into thinking back to like grade school when you guys learned about like the food chain and the like the exchange of energy and carbon sources basically in the food pyramid. I don't mean the food pyramid like bread and vegetables. I mean like, you know, you have your plants at the bottom and the things that eat the plants and the things that eat the plants and the, you know, predators and stuff like that. Yes, you get what I'm saying. Okay. Anyways, it's in our textbook, but I figure we've heard about it enough and I don't care about it enough to go over it more. So we're focusing on the fancy stuff for now. All right, everybody tracking on this so far? I hope. Okay, so I'm going to leave that up for now. Good. Okay, saprobes. What do saprobes do? So Jeremy, you're going to be our next victim. Ask you a question about this. So saprobes break down dead things. We call dead things detritus or detritus. I don't care how you pronounce the word as long as you can recognize it on an exam. So we think of like fungi being a primary saprobe, one of the major saprobes. We have plenty of bacteria that help with breaking down dead things as well. They're also saprobes. What I want to point out about saprobes are breaking down organisms, right? They're actually using the organic matter from organisms. They're not living organisms now. They're dead and decaying organisms. And so saprobes are not autotrophs. They are not photo anything. Not photo, they're not autotrophs. They're not chemoautotrophs. Okay, so what is the word, another word that we might use for them? From one of the terms that we have up here. If these are the four categories that we generally fall into, if they're using organic matter, so heterotrophs. Saprobes are heterotrophs. So that's okay. We'll come back to. That was a trickier question. That's not on you. A lot of us were kind of getting tricked by that one a little bit. So saprobes are heterotrophs. Yeah. So they're still getting their organic matter from some other organism that made organic matter. It's just, it happens to be a dead thing. The things that we eat, we think of them as being dead, but they're not dead and decaying yet. We don't let them be dead and decaying, yes. So we're not really there. We're like predators. We like to fancy ourselves that we're not eating live things that we just killed and just gnawing into them. But we are what we consider omnivores, right? So the plants that we eat are technically still alive. Unless you cook them, then they might be dead. And that just changes the protein forms to be a more digestible version of the proteins. So it's just different levels of that same kind of thinking. We're heterotrophs, but we're not saprobes. We're not only feeding on dead, decaying things, right? So saprobes are still heterotrophs, though. So weird one. That was a tricky one. Don't worry. If you didn't get that one right away, we'll give you an easier one next. All right, next question, question 4. Diffusion of water molecules through the cell membrane down or with the concentration gradient. We're moving with a concentration gradient. Water is what's being diffused. And it's across a semi-permeable or selectively permeable membrane. All right, so this is osmosis. Anytime water is diffusing across a selectively permeable membrane, that's the definition of osmosis. All right. You want to ask about any of this? Can you tell me anything about active transport or facilitated diffusion? Jeremy? Either of them. Or even exocytosis. I'm not going to be picky. Any of these other 3. Can you tell me about what they are? All right, we'll come back to it. That's all right. No worries. So think on that one. When you have an answer, let me know. Okay. All right. Beautiful. I'm going to clear this for now, and we're going to move on to the next question. It might be in the next question. I don't know. All right. What is the solute in salt water? You know, I have an easier question for you. I'm going to ask you what the solvent is for this. Hopefully that'll be, well, I'll let you choose what you want to answer. We'll give you something. Solute and solvent are different things. So what is the solute in salt water? Okay, so solute and solvent. This is, we clearly need to work on it. We need to work on it. So that's a term that I know we've had it before, but we need to get them cleared up in our brains, right? So if the solute in the water is salt, what's the solvent? Okay, fair enough. That's the easy enough one, so I'll give you credit for that one. But yes, so that is one we need to get kind of straight. I don't know how to make it clearer for folks. To me, when I think solvent, the thing that is dissolving the thing, but then other people are like the thing that is dissolving in the thing. And then I'm like, okay, I don't want to confuse you. So I don't know how to help you out with remembering solute and solvent. And so I have to think of both of them at the same time to know which is which. Honestly, because if I'm just seeing the word solute like we did on here, and I haven't talked about solutes and solvents in a long time in solutions, and even for me, I'll be like solute and solvent, which one was which? And I'll get solvent. And for some reason, my brain knows that the thing you're dissolving in is the solvent. I don't know why. Maybe organic chemistry. I don't know, because we had so many solvents, and often we used acetone as a solvent, and it was the thing that we would dissolve organic molecules in, because they don't dissolve in water, right? They're nonpolar. So I don't know, but maybe that's why that stuck with me. I don't know how to help you with it, but definitely work on knowing solute and solvent, because you know it's going to be on the test, right? So, okay, we'll do one more question from here. Will be last one for chapter 9. Solute concentration outside the cell is the same as solute concentration inside of the cell. What is that? Which one would represent? Same inside as outside. So Danielle. Oh, right, okay, yes. I was like, you know, you sit in the back. I was like, yeah. All right, so this is isotonic when they're equal. So can you explain in your own words? You don't have to use solute and solvent or whatever if you don't want to. You can say salt concentration or whatever. Whatever works for you. Can you explain what a hypertonic solution would be? Hypertonic. Uh-huh.
Higher.
Yes, so solute, so to put it in perspective, in case we're still having issues remembering solute and solvent, the salt concentration would be higher outside of the cell. It's not always salt. So we often think of salt as being our comparison, but even sugar. Sugar could be an example of that. And it could be anything, honestly. But when we start talking about preventing microbial growth in things, we often think about how having high salt concentration outside of something, and you pack something in salt, it draws the moisture out of the cells, right? And we know that because of this hypertonic, hypotonic situation and osmosis that's involved there. So we know packing something in salt draws the water out of the cells and then can prevent the microbes from growing and even kill them. You pack something in sugar, it'll do the same thing. So ever think about that with preserves, with jam, with things like that that are high in sugar. That's why they last so long on the shelves, is because they're high in sugar and it has that same effect. We call that osmotic pressure. So that's going to come back around in unit 3. All right, beautiful. Okay, so we got Daniel. Okay, so that'll be our last one for this chapter. So I'm going to move on to chapter 25 briefly. Luckily, chapter 25, we already talked about the nitrogen cycle, so we don't need to cover that. Okay, how do I get out of this? Here's our podium for this one. Just so you guys can feel good about yourselves for a second, before we get too far into chapter 10 and start feeling overwhelmed. All right, good. All right, we'll do chapter 25 next. Okay, chapter 25, slide. Make sure we have the email thing on. All right, we'll sign back in. Give you guys a minute. And give you a minute to pull up your chapter 25 notes if you need to. 1, 2, 3, 4, 5, 6, 7. We've got 7 people down. I want to get 3 more people from this chapter. So what we talked about in chapter 25, we're going to be talking about the carbon cycle, the nitrogen cycle, nutrient cycles in general. We're talking about water treatment, whether that is water purification or like drinking water or wastewater treatment, and also One Health. It's a brief concept. I just need you to know what One Health is, because I feel like a lot of what we talk about, we're focusing on how bad microbes are for us, but we need to think about the fact that microbes contribute good things to our environment and that the overall health of our environment affects our health too. And that's the idea of One Health. We might have just said enough about it that we don't need to cover it, but yeah, I mean, it's just the big picture look at things. All right. We're going to go ahead and get started. It seems like we still have 1 person that's not signed in. That's okay. We're going to get going. Remember, you can always sign in with that code at the bottom of the screen. Let me move this out of the way. Some blank is a more potent greenhouse gas than carbon dioxide. You want to talk about what's harming our environment, what's causing global warming. Again, if you haven't looked at the actual trend of global average temperature, actually like looked at it mapped out on a graph, it's inarguable that the global temperature is going up. Okay, now carbon dioxide is one of the major contributors to that temperature rise. We know it acts as a blanket and keeps things warm, but something that is even more potent, even worse for our environment is methane, CH4. And if we know our major source of methane here in the United States is our agriculture industry. So probably one of the biggest contributors to global warming. We think about industry as being a problem, and it is industry, but probably not what you think. It's probably not mechanical industry and cars and things like this. I'm not saying that they aren't causing, problems to our environment, but the livestock industry, massive. I mean, all the farts from all the cows.
It's so bad.
It sounds ridiculous, but it's a huge problem. And if you really look at like farmlands and like planting crops as close together as they are, and you look at like the effect of having that, that has caused massive impact in environment as well. There's such the amount of heat that the crops give off from being growing that close together. You might think like, oh, that land would be having plants in it too. Yeah, but not big, fruit producing plants that are that close together like that. And so the heat that they're giving off is also contributing significantly, changing the climates of the local areas. So yes.
Carolyn, you have a video for this?
Mhm.
The subject got flagged for that. Talking about-
We're talking about global warming.
Yeah,
I
I just signed into my YouTube to watch it.
So, yeah, if you have to do that on YouTube, then I guess that's the thing. So it gets also flagged too when I play like video recordings. Sometimes I do a video recordings from McGraw-Hill that I'll play like during the lecture, and it flags my videos for that because I don't own the rights to those videos. I'm just trying to record my lecture and put it online so that you guys can watch it, but yeah. So, and also sometimes too, whenever I'll talk about like people think blah, 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 and it thinks that I'm saying blah, blah, blah. And then it'll be like, you're talking incorrectly about a concept. I'm like, I wasn't, I was quoting other people for it. So I've gotten quoted for that with the autism vaccine stuff in the past, because I'm quoting what they used to say about it. And then the YouTube video flagged it. And I'm like, actually watch the video. And so anyways, so yes. So, just FYI, probably methane is probably the biggest producer, probably the biggest issue with it right now. I'm going to go on to the next question, so I'm going to get stuck on this. All right. Plastic creates islands in the ocean that can cause serious shifts in the microbial balance. I don't know what questions to ask about this stuff. To me, it seems pretty basic. I don't have a lot to say about it. All right, so this is true, obviously. I mean, if you have big islands, and there's been shown, and it might be bigger now, but at the time that this textbook was written, there was an island the size of Texas that was out in the Pacific Ocean, kind of south of India, that was just plastic. So that is massive, and that's a huge problem. Obviously, that's blocking out the light, so we're not getting as much sunlight reaching through. It's creating surfaces for microbes to create biofilms on, and then they're blocking out more light, or they're using up nutrients in the area that they otherwise wouldn't be using up because now they're, attached to this plastic and whatever. So that creates these weird like microbiomes in the ocean that just significantly affects the ability of organisms to live in those areas. During photosynthesis, this is talking about the carbon cycle here now. I mean, yes, this is a picture of it, but I don't know how much you can make out of it. All right, so during photosynthesis specifically, we are taking CO2, which is inorganic, using the sun. Essentially, this is the watered down version. Obviously, in chapter 10, we have 2 steps involved in this, but carbon dioxide plus sunlight to make organic molecules. That's the idea of how the general overview of how photosynthesis works. Now, there's 2 steps to this, the light cycle and the light independent cycle. And so the light cycle, we just call it the light dependent cycle. And then the light independent cycle, we call it the Calvin cycle. So there's 2 steps to this, but that's the idea. So we are converting carbon in carbon dioxide form from that gas form into carbohydrates in the form of glucose. And that's the idea of what's happening in photosynthesis. During aerobic respiration, just kind of getting into carbon cycle, but also getting into chapter 10 a little bit. Does anybody who hasn't answered a question yet, or anybody want to tell me, where does carbon dioxide come from? Which step in aerobic respiration? Anybody that hasn't answered a question, no. So we have 3 steps in aerobic respiration. We kind of have 4 a little bit. We have glycolysis. We have an intermediate step, which is why I say we have the Krebs and we have the electron transport chain. And this is cellular respiration in general. In aerobic respiration, we're using oxygen as our final electron acceptor in the electron transport chain, right? So in which of these steps, and it is more than one of them, is carbon dioxide produced? Anybody that hasn't answered a question yet? Lauren, saw your hand up first. Yes, so 2 and 3, intermediate and Krebs. We make carbon dioxide in both of these steps. Beautiful, perfect. Okay, just leading into that so that we can keep that in mind from this stuff. So I think this is a good point to say, let's start 10. Sound good, everybody? I know we didn't get into symbiosis yet. Does anybody have any questions about symbiosis? So remember, just really quick to review symbiosis. Symbiosis is just a relationship between two organisms that at least one of the organisms depends on that relationship to live. It's not an optional thing, okay? So that's symbiosis. Good and bad things can happen in symbiosis. There's 3 different subtypes of symbiosis. In commensalism, one organism benefits from the relationship and the other one is neither harmed nor benefiting. It's just there, okay? In a parasitic relationship, we got Lauren already. Let's say London McNair. London McNair. It's London McNair here because you're Cooper, right? Okay, London McNair. Okay, so you're London McNair. Okay. Parasite. Can you tell me how that symbiotic relationship works? Parasite, just generally. So I'll give you our hint. I'll write it about here so we can have some reference. There's 3 types of symbiosis. And I covered commensalism. The other 2, one of them is parasitism, a parasitic relationship. There's 2 organisms involved. Can you tell me what's happening with the 2 organisms? Yes, so we will say the host is harmed and the parasite benefits. This is general, specific, like we can think of this as general speaking, right? When you think of a leech, that's what happens, right? When we think of a virus, that's what happens. All right, so that's general theme here. That's what symbiotic relationships are. And then the last one, anybody, let's see, actually, let me call on a person here. Kay Sanders, are you here today? Kay Sanders, not here today. Jordan, we already got you. Jackie Malfa. I don't know who we have online either. Sammy. I guess I'll ask Sammy just because you're here. Sammy, because I just happened to see your name. And I know I haven't called on you yet, and yours is at the end. So, but anyways, so we'll just call on you now. What is the last of the symbiotic relationships called? What is the term for the last of them? And I'll help you out with what happens with it. Both organisms benefit. So Sammy, if you can tell me in what type of symbiotic relationship do both organisms benefit? What is the term? And I'll let you answer that. You can write it in the chat or say it over the speaker. And then I think we'll leave that there. Any questions about any of that for everybody? Mutualism, perfect. All right, we good? Okay, let me just delete everything then. All right, beautiful. Right, here's our little podium again. Yes, quorum sensing. Okay, so quorum sensing is literally just a way for organisms in a biofilm to communicate with one another. So really quick, while we have this screen up, I'll kind of draw out a biofilm. So you might have bacteria that have capsules, right? There's capsules or they've got slime layers, things that help them stick to a surface, come in and they're sticky and they're gloopy and they're sticking onto a surface. Now, other bacteria can come in and stick onto that. And then other things can come onto that, including fungi. Here's a fungi, and here's some fungi, and whatever. And we're all getting all glooped up together and whatever. So we're creating this biofilm. This is an example. It's many layers thick, and it can be fungi or bacteria, or even sometimes protozoa.
Is that initial one the pioneer pollinators?
Sure, but you don't have to know that for my class.
Oh.
Right, so that's not in my lecture, so you don't have to know, but yes. Okay, so yes. I won't test you on that, but that is true. Okay, so this is a biofilm. Now imagine that somebody's coming in and brushing their teeth, okay? Somebody's coming in and they're brushing their teeth, and these guys start dying, and they're releasing parts of their cells, the insides of their cells, like their nucleic acids and things like that. They're supposed to be inside of them, and they're now outside of them. So now imagine we have signal molecules. We call them signal molecules, but they're just molecules from dead cells that the other organisms in the biofilm can basically sense them. So they are saying there's DNA around here. That's supposed to be inside of a cell. I maybe don't want to be in this biofilm anymore. That's quorum sensing. So there's that simple kinds of stuff. There's also chemical signals that they can send to one another. Like if I find some good food, I might send out signal molecules that let other bacteria know good food is in my area. And then they can track my signal to where the good food is. So it can be more specific too. We have general, like if I find cookies on the table here, imagine me eating the cookies. I mean, this is getting crass a little bit, but if I'm eating and it's making me fart, and you guys are like, somebody's farting, they must be eating something, and that's making them poop or fart or whatever, you know, they're making waste products. So then you guys sitting around or nearby or in the biofilm too, you'd be like, somebody's eating because I can smell their waste. So let's go towards them and go see that kind of waste, meaning there's food somewhere near us. Other kinds of inside of cell products, we sense that happening. We might be like, oh, that's a bad thing, we want to leave. So you can kind of sense the environment based on what organisms around you are producing as waste products or even signal molecules. We do not need to worry about what the exact signal molecules are, but they'd be like proteins and things like that, that they would be making as a result of finding food or something like that, or sensing danger. If I am at the outside of a biofilm and I sense a chemical that I know is dangerous, I might even make certain signals that let my nearby, like a skunk makes a bad smell to let their fellow skunks know something bad is attacking them and to ward off the bad thing. Bacteria can do the same thing. And then bacteria's ability to sense what is going on in their environment like that. Is there good stuff happening or bad stuff happening? That is quorum sensing. Okay, did you have a question about it?
That was only in bacteria.
So bacteria, any microorganism in that colony can do that.
Okay,
So
like
we
it
talk
has
about
to
it
be
more
biofilm.
specifically with bacteria.
Okay.
It's more common. Almost always when we have a biofilm like this, it's going to be involving bacteria. Yeah. But it can be bacteria, fungi, or protozoa. That make a little more sense? It's a more general idea of responding to signals. It's not like necessarily specific signaling, though there can be specific signaling, even general response to your environment and then responding to it.
It's like bacterial survival instinct.
Yep, that's it. So we think of it as so simple because we think with brains, right? That whenever you smell cookies, you want to walk towards the cookies. For the bacteria, they have to pick up on the chemical signals and then their cells have to respond accordingly to move towards or away from something in their environment. Now, if we're just one bacteria and we're sensing that we want to move towards something, then that would be chemotaxis, right? Remember we talked about that motility wise. So that would be chemotaxis. But the same concept works within a biofilm. So now we're a big community and we're living all together. And let's say you're living in a building and you smell smoke. Think of the building as the biofilm. When you're smelling smoke, you might want to leave the building. It's the same thing with quorum sensing. And bacteria smell smoke and it's biofilm, it wants to leave. If it sees DNA and stuff that's supposed to be inside of a cell floating around in its little biofilm, then it might want to leave. senses anything. So it's a similar idea to the chemotaxis, but instead of moving to or away from something in motility, now we're talking about being part of or moving away from a biofilm community. Makes sense. That's what form sensing is. Does that clarify that a little bit better? Okay. All right. So, okay. So now really briefly, we did chapter 9 and we did chapter 25. We have the activities for these and together they're 5 questions. So we're going to go through those activities really quick. I'm trying to click on this and it's like weird. All right. Chapter 9 activity. This is just basic stuff. There's no case study for this one. All right. So just general questions. What is the difference between diffusion and osmosis? Kay, we already said Kay isn't here today, right? I think we agreed on that. Kay is not here. Jackie wasn't here either. Okay. Catherine Fields. Catherine. Okay, Catherine. Do you know the difference between diffusion and osmosis? So that's good. So we'll start there. So I'll give you credit enough for that. So don't confuse yourselves on this, right? Osmosis is the diffusion of water. It's diffusion of water specifically. Okay. So when we diffuse, if I had, again, I talk about this in the lecture, but this is a good example. If I had a big old pile of bouncy balls here, like the beach ball kind, right? And then I just like let them go, they would roll around through the room, spread around the room, right? That's diffusion. So diffusion is anything moving from an area of high concentration to low concentration. And physics says that they want to spread out. That's physics, not chemistry. Physics says that. So physics says molecules want to spread out evenly everywhere. And if you remember when you learn about the laws of thermodynamics, then that is what we would call entropy. It's the law of entropy. It's the idea that everything is going to a state of basically breaking down further and further and further to be more simple and simple and simple and not as organized and structured. That really is the gist of what entropy is. And so things don't want to be piled up in a big pile. They want to spread out evenly everywhere. And the entire universe is moving towards that tendency, by the way. And if you're going anytime against that, you have to put in energy in order to fight against what the universe naturally wants to have happen. And so diffusion is movement of molecules from high to low. Osmosis is diffusion of water specifically. Okay? And it's usually dependent on that selectively permeable membrane. So that's fine. I'll allow that one. All right, our next question. Saline is often administered to patients to keep them hydrated. We talked about this way back in the beginning of class, but saline is often administered to patients to keep them hydrated. Do you think saline is hypotonic, hypertonic, or isotonic compared to a patient's blood? And defend your answer. So here, I'll help you out a little bit. We want isotonic. We want isotonic compared to a healthy person's blood, not to your specific patient's blood. To a healthy person, you would have 0.9% sodium chloride. We have that amount of solution there. If we have pure water, if we were to, I guess that's what the next question is, right? Question 3. So I'm going to ask Amna this question. If we were to give an IV to a patient of pure water, what would it do to their cells? It would make them burst, right? Because that's exactly, that would be a hypotonic solution. And so the red blood cells and the cells in their capillaries and everything like that would try to take up more water to water themselves contents down to try to make it equal on both sides, but they can't because pure water is pure. And so those cells would swell up and burst. And that would kill you. If you give somebody enough pure water, get a little bit, your body will handle it. But if you give enough, you give a full liter of pure water to somebody, that's going to kill them. So that's why we give saline, so that we can keep that isotonic environment. So that if somebody is dehydrated, their blood might be hypertonic, but we want them to bring them back to that isotonic quality and make sure everybody's getting hydrated. So the healthy, aspect of your kidneys isn't getting taxed too much. Because we all probably, if you have taken anatomy or physio and or both of them, then you learn quite quickly that your kidneys are what is maintaining the balance of your blood, period. So they're maintaining how much salt concentration is in your blood, the buffering of your blood and everything like that. But your kidneys are managing that. And so they're doing quite a lot for us. let alone filtering out waste and stuff like that. So if all that stuff is being managed at the kidney level, that's gonna be managed there. And your kidneys can only manage so much. We get enough pure water in there, now we're gonna have bursting of our red blood cells, and now your spleen is gonna be dealing with the ruptured red blood cells. And that quickly will also signal to your body, we're having ruptured red blood cells, alarm systems will go off, and that will cause shock in the body. And we know we will have hypotension and things like that. That can lead to death pretty quickly if it's not managed quickly. That makes sense? Okay, I know that is more of a physiology question, but it is, I feel like, a learning opportunity. So there's that. All right, so chapter 25, we did have a little bit of a case study. It's talking about COVID. Remember the virus that causes COVID, SARS-CoV-2. SARS-CoV-2 in wastewater. So this is talking about challenges for developing countries in managing SARS-CoV-2, COVID basically. So the review mentions that SARS-CoV-2, the virus that causes COVID-19, is found in high loads in urine and feces. Why would this be a public health concern? So obviously, if we are pooping and peeing out that virus, right, and then all that waste is going into the water, where in third world countries and underdeveloped countries, they are systems in place. They have no systems at all typically for managing their waste and things like that. So they are also washing and bathing and getting their drinking water from the same source that a lot of their waste will run off into. So now they're just contaminating their people. And if you don't think this is a problem, it is literally the reason for why they have helminth infections and we don't, because we have wastewater treatment and they don't. And so a lot of those helminth infections are either, we obtain them either through fecal oral contamination or from walking or swimming in fecal or urine contaminated waters. That's why they have helminth infections and we don't. So that's that idea of that One Health, and picturing not just how we're living our lives here in America, but in keeping in mind, the health of the whole world, not just people, of course, but also all of the environment and the animals in it. Fecal coliform testing. Let's see if we can get this one. Is Kaya here today? Kaya? I don't think so. Okay, Kaylee Jackson. Okay, so fecal coliform testing. Fecal coliform, so I'll give you a little bit of a reminder of fecal coliforms, yes.
No.
Oh, okay. So fecal coliforms, they are bacteria, okay? This is saying that if I see E. coli, Klebsiella, Gardnerella, normal poop bacteria in our treated water samples, if I see those there, then things like Salmonella or bad E. coli, not normally E. coli, but the bad E. coli, or Shigella, and those things could be present in our water treatment, in our water as well. The normal guys are there, then the pathogens could be there. So fecal coliforms are what we consider indicator bacteria. Okay, so that's my question for you, which is this question. Would that be helpful for testing for SARS or SARS-CoV-2 or COVID? Defend your answer.
Well, I just feel like with everything that's in, I don't know.
Okay. So I get where your line of thinking is going. So I'm going to give you credit for thinking through this, but the answer is no. And this is why. Viruses are much smaller than bacteria. So just because you could have no coliforms, completely coliform free, but every virus in the entire universe can still be getting through. They are that much smaller. So keep that in mind. Viruses are much smaller. This is a filtration process. Okay. So water treatment relies on filtering things out. So this was a problem in polio, in the days of polio. When we had polio, polio is a disease, to be clear, that only exists in people. Animals don't carry polio. Only people have polio. And it was fecal oral transmission. And so they had to devise a way of wastewater treatment that eliminated polio specifically. And they did. We had to reintroduce that concept of how we eliminated polio back in whenever we started seeing COVID, because that was the only thing that we had in place to keep, because we don't have polio in the US anymore. It's been eliminated from the US. We don't need to treat our water for it anymore. But we had to go back to that line of thinking in order to think about how we were going to be treating again for this. So vaccination eliminated it and we didn't need to filter it out anymore. That's how we got rid of polio. But obviously COVID, it's not the same. So Yes.
How does it not just restore?
Polio?
Yeah.
I mean, it's literally, we don't have it in the US. The only place in the world that they have it still is Afghanistan, Pakistan, in like very rural areas. So those people don't come here with the polio. Typically also, if you have polio, are you running around from country to country? Right, right. Typically ain't running around at all. Typically, right? It's a very debilitating disease. So you will be paralyzed typically from polio. Even if it's temporary, it's usually what we would see. So that's why we don't see it here. If that answers that question. Any other questions about this stuff? All right, so water treatment stuff, what I want you guys to get out of it are the basic steps of it that I go over in the lecture. We don't need to know like every single little detail about the water treatment, but have the idea in your mind that we're gonna remove the big heavy things first, the solid waste first, and we're gonna have the sludge left over that we will let bacteria usually biodegrade the sludge, and we can reintroduce whatever liquid is left over from that back into the whole filtering process. So we filter it through several levels of filtering, and then we will usually treat it chemically or with UV treatment as well before we release it back into the environment. And yes, we release your pooped water back into the environment, typically into lakes, rivers, or like golf courses and stuff like that. We can use that water for watering golf courses, parks, and stuff like that. There's different acceptability for how many coliforms you can find in drinking water versus like water that you would use to water a park versus water that we release back into the environment. How many coliforms can we find in each of those? Obviously, drinking water, we want to get as close to 0 as possible, but there is some allowance there. So there's different ranges there, and it just depends on what you're using the water for. Any questions about that? Cool. All right. That brings us back to our Kahoot. So we're going to reset here really quick. We have some. Yeah. You're good.
This could be wrong, but I've heard like certain things aren't as easily broken down or cleaned out of water, like hormones, like hormonal birth control and things like that.
Uh-huh.
Is that possible that that's like accidentally cycled back through after the water monitoring and everything?
So unlikely. It's unlikely just because like we treat the water and filter it and everything and then release it back into the environment. We don't release it straight back into drinking, right? We release it back into the environment and then you might get your water from that environment again, but it goes through the drinking water filtration process on top of it, which is even more stringent. And then there's enough filtering in that and even things like reverse osmosis and stuff like that, that we are removing the chemicals and stuff like that out of the water. So much so that oftentimes we have to re-add back in minerals after treating drinking water. Yeah.
That's what I've heard, that like reverse osmosis is the healthiest thing you can do
Yeah.
because it filters it more and
Mhm.
you can remineralize it.
Yes. Yes.
I will say that because all the submarines do make our own water,
Mhm.
and you have to put minerals in it, or you will sit there and drink and it will not hydrate you.
And that's what I
It's pure.
I went to, so I was at Atwoods and we were trying to find water softener. So we have an RO system at my house. So I live in Bethany. Bethany has the worst water in like the state of Oklahoma. Just to be, I'm not even kidding. Like it literally is documented as being almost borderline illegal how bad Bethany's water is. If you live in Bethany or have family that lives in Bethany, do not drink the tap water. I'm being serious, okay? Bethany's water is terrible. So I have a reverse osmosis system at my house. It wasn't cheap, but I feel like I'm paying for it now, like avoiding things.
Like you don't have to worry about.
Yes.
that at all.
And Bethany buys some of their water from Oklahoma City, but they also have their own water sources. And they've had so many like boil water like notices that you'll have to have. Like it's unreal. I feel like I'm living back in like 30s or something like that. Unreal. But anyway, so I have reverse osmosis. But anyways, I went to Atwoods to go buy water softener. And the lady was like, I don't even understand what water softener is, like if your water is hard or whatever. I was like, so my water is pure water. And unless I add back in minerals back into it, and your body, like we've already said, if you give somebody pure water as an IV solution, that would be bad for their body. And it's the same thing in the water that you are drinking, in the water that you're showering in and everything. So you have to add some mineralization back into it. And that's how RO water works. So they can do that too with tap water purification for citywide treatment. It's not gonna be as high of scale, but it's a similar concept with that too. So, but yes, that's how RO systems will work. It's the cleanest water that you can get. And then we also have another filter at our like tap. So when I bring my water to drink like this, it went through my RO system and a filtration system before it comes out of my tap. And
Are
then
you having to change your filters constantly?
no, it's permanent. Yeah, so it uses like, physical removal of things, but it also is, I think UV and stuff like that to kill microbes. It's a UV filtration system. But the RO system is literally like just use chemistry to like pull anything that's not water out of the solution. It's osmosis, but it's reverse osmosis. So it's pulling anything that's not water out and then leaves the water behind. And that, anyways, then that flows through the pipes of your house, which are probably not clean either, just to be clear, right? So when we think about drinking tap water, do you also think about the miles of pipes that your water has flowed through to get to your house? Exactly. Me too. So that's why I only drink the water that comes from my purified water tap that has like a, brand new line and a new tap on it. And I don't drink my tap water either because my house was built in like 63 and it has, cast iron pipes or whatever. And it's
Lovely.
yeah. Anyways.
So high maintenance just to be healthy.
Yeah, you don't think about this stuff. And I'm not saying that like that means you shouldn't drink the tap water at your house. That's not what I'm saying. If you live in Bethany, you don't. If you have well water, don't. So if you have well water, I also wouldn't drink that. I would get a reverse osmosis system for sure if you have well water. But otherwise tap water from the city system, it should be fine. They're testing it and they're making sure they're considering it flowing through people's pipes and stuff like that. Okay. If you want to drink purified water, don't let me stop you either, because obviously I do. But I purify mine. If you drink bottled water, then, that's just as good. So.
There's some days where like you turn on our house, it's like a swimming pool
Mhm.
or straight beach.
Yeah, some people were having issues with that in Yukon lately. I saw people posting about Yukon happenings. Mhm. Yeah. But that chlorine smell that you get sometimes in that tap water, that's from chlorine that they use to treat that water. There's an allowable amount of chlorine, and Bethany adds so much chlorine to their water. It is absurd. Like whenever we were testing it, we were testing before the reverse osmosis and then after the reverse osmosis, and you could smell the difference in the water.
Wouldn't that be fire on your butt?
Anyways, so I'm not telling you guys how to live your life with your water, really, honestly. I'm just telling you how I live my life with my water. And so, yeah, you do you though. Okay, we're gonna get in chapter 10. I want to get in some of the spicy stuff here. Where are we at? We do have London, Cooper, and East. We only got a few people left. London, and then Taylor, and then John. All right, which term refers to all the chemical reactions that occur in the cell? All chemical reactions, breaking down, building. There's one term whenever they are all. One of these terms is like the other one, but they are not the same. All right, so this is metabolism. London, what is catabolism?
Like the breakdown of all.
Beautiful. Breaking down, exactly. Breaking down molecules to release the energy. Anabolism, like we think of anabolic steroids. Anabolic steroids are the steroids that you take to build muscle, building. Okay. You build things by adding in energy. So anytime you're making a new thing, you need to add energy to it. Amphibolism. Amphibolism is a term referring to the linking together of catabolic reactions to allow anabolic ones to occur. So metabolic reactions, we're talking about just all of them together, but if you're talking about amphibolism, this is the idea that you need to break things down to get energy to then build things. So it's just a little bit more specific. Okay. Beautiful. Next. Let me flicking through this. Okay, anabolism. Well, see if you guys are paying attention. Get through this one really quick. I need some gum. Well, I'm drinking my water. All righty. Anabolism. Building molecules by using energy. All right. So catabolism, cats destroy things. Catabolism is breaking things down to release energy. Anabolism is using that energy to build new molecules. We have to use that molecule. So that's the difference here with number 6. If we read through, do we build the molecules and release the energy? No, we need energy to build. And the way I always think about this is you're building something like a log cabin. You need a lot of energy to build a log cabin. I frame it that way. It's the same thing for your cells too. They need a lot of energy to build things. So, next question. We are on chapter 10, Jackie. And we'll probably call on you in a little bit. Not right away, but I'll give you some time to settle. Enzymes are an example of what macromolecule? All right, so enzymes are proteins. So we'll think of this as anything with ACE at the end. So DNA polymerase, DNA ligase, collagenase, anything ACE at the end of it, they're generally enzymes, and enzymes are generally proteins. For our purposes in this class, we're only going to talk about protein enzymes. So don't even worry about any other kind that there could be, because it's the proteins. Okay. All right, next question. Where does the substrate bind on an enzyme? Right, so this is called the active site. So remember, enzymes are proteins that help lower the activation energy of a chemical reaction. So what this means is, if there's an enzyme that breaks down glucose into, I don't know, what was that first one in the glucose 6-phosphate or whatever. It adds on a phosphate on there. We remember glycolysis, and there's like the many steps that you guys don't need to memorize. But let's say we're breaking down glucose into pyruvate, and there's an enzyme in there. There is, but I'm not gonna make you know what it is, okay? That enzyme acts on whatever it's acting on by binding it at that active site there. I'm trying to say, I had a whole thing in mind, and then I went off the rails there. Right, so breaking down glucose to pyruvate. There's a lot of steps involved, but that could happen in physics, I'm talking about, okay? Naturally. If you just have a glucose molecule, could it just naturally, spontaneously break down into pyruvate? Technically, yes. But what are the odds of that happening on their own without putting some energy in there? Then you put a lot of energy in there. The amount of energy that you would need to put in there to break down glucose to pyruvate would be a lot. By using enzymes, now the enzymes just help that stuff go along, so you need less energy to make it happen. That's what a catalyst does, okay? And so the enzymes themselves are not being permanently changed. They can revert back and forth and be reused over and over and over again, like DNA polymerase. It's a little enzyme that adds on the next T, the next A, the next G, the next C. And it's being reused every single time as it moves on down that chain while we're going through DNA replication, for example. Okay. All right, so the substrate binds, here's a little pizza shaped guy. He binds at that active site. All right, so let's see if it gets more complicated than this, because I can ask a question about it. Because you guys know what's coming next is conjugated enzymes, hopefully. Okay, nope. What are causes of a denatured protein? What can cause proteins to denature? All right, so pH, heat, and chemicals. So I posted this for my online course, but I can give you guys a little bit of a talk about it in person. When we talk about proteins, they are amino acids linked together in a big old chain. We know that this is made during translation, right? Based on whatever your mRNA codon is, we know which amino acid goes where. All right, this is our primary structure of our protein. Then, based on whatever your R group is on your amino acid, we can interact with the guy near us, or later on down the chain with the guy across from us. Okay, and then this leads to our basically let me erase this before I get too confused. You get the idea though. This would lead to our alpha helices, which again would just be amino acid here, amino acid here, amino acid here, whatever, and how they're interacting, the R groups are interacting, or our beta pleated sheets, which would be amino acids like this, and then a little turn, and then amino acids like this. And then these guys might interact with each other like through hydrogen bonds like this. This is our secondary structure of our protein. Then, I know this is chapter 2 stuff, but you can't tell me that this doesn't relate directly to what is going on here, right? So our tertiary one is where you had your alpha helices and beta pleated sheets. And now the way that they want to interact with one another and make up that folded structure like this, we might have hydrogen bonds across the way. We might have disulfide bonds where there's an S and an S. We might have ionic bonds as well, just depending on what types of amino acids we have there. And so disulfide bonds, disulfide, hydrogen bonds, or ionic create this structure at the tertiary level. All right. Now, how much heat or involvement of extra H pluses? So let's add heat. That's what the delta is. Increase heat or add extra H pluses, increase your H plus concentration, or add certain chemicals in there. And now you can break your disulfide bonds. First, your H bonds, those are going to be the weakest ones, right? Then your ionic bonds, the next weakest, and then your disulfide bonds, which are covalent bonds, but you can still break them with the right chemicals. So pH, heat, and chemicals can break down that tertiary structure, even your secondary structure, and then just leave you with your primary. How is that going to function? Whenever we needed this shape to bind onto our substrate, now we've ruined that shape. You get what we're saying with denaturing? So heat, extra H pluses, which is pH change, or too few H pluses, which is high pH. Any of that can alter our ability to make any of these bonds here. It can affect that severely, and then lead to denaturing. So that is basically destroying the shape and therefore the function. Oftentimes in enzymes, the shape is more important than the exact amino acid sequence. Amino acid sequence is still important, but often that is really key is the shape, okay? So that's how denaturing works. Everybody following that? Okay. Next question. Still got some questions that I want to ask you guys, but I just want to be sure that we got our enzymes down. I don't know if it's going to talk about enzyme inhibition yet. Hopefully it does. What best describes non-competitive inhibition? Non-competitive. All right, so if this is our enzyme in its normal state, and normally it binds onto our substrate like this, and in a non-competitive, this was terrible. In non-competitive, what will happen is something might bind here, and then that causes our enzyme to change shape to this. And now this can't fit into there anymore. That is non-competitive. Did not bind at the site where our substrate binds. So Jackie, I'm going to ask you a question about this one. If our inhibitor bound at the site where the substrate binds, the active site, if the inhibitor bound there instead, what type of inhibition would that be? It's not non-competitive because obviously non-competitive binds somewhere else. It's not competing with the substrate. So if we're competing with the substrate, what type of inhibition would that be? Now imagine that our inhibitor looked like this. And it's got some spikes coming out, who knows? All right, I'll let you cook on that. Next question. The 5-carbon sugar found in ATP. Righty, so the 5-carbon sugar of ATP is ribose. So this would be your ribose. And then we are linked to adenine, which is a nitrogenous base. 5-carbon sugar linked to adenine, and then we're also linked to PO4, PO4, PO4. And that is basically what ATP is, right? So ATP is our energy forming molecule for the cell, all right? So our next person is Taylor. How is ATP storing energy?
Isn't it to do with like the breakdown of ATP and using, like breaking that down and using it for energy?
Yes, but how does it have energy? Like breaking it down, what releases the energy from this?
Okay, so it's adenosine plus the extra phosphate. And so is it the extra phosphate being used?
Yes, so close enough. Okay, so we're getting there. So yes, so it is literally the phosphate. So each of these phosphates has a negative charge to it. All of them do. And what do you guys know about if you put a bunch of South Pole magnets together, they repel each other. North and South will attract each other, opposites attract. But if we put the same charges right next to each other and we are covalently bonding them to each other, these guys are repelling each other constantly and they want to leave. So there's so much energy held in this bond right here between the second and the third phosphate here that whenever we break that bond there, breaking that bond is what releases the energy from ATP. So it's in the phosphates. Yes, it's specifically because the phosphates are so negatively charged, they want to get away from each other. That's what it makes ATP the energy source for the cell. Okay, we still cooking, Jackie?
Oh, I found it.
Okay, sorry. Let me know whenever you get that. So whenever the inhibitor binds at the active site, what type of inhibition is that? It's not non-competitive, it's the other one.
Oh, right, that's right.
Okay, so that is where, so I'll give you credit for it because we're in the realm of that's what we're talking about and I don't want to spend too much more time on it. But in non-competitive inhibition, when your inhibitor binds at somewhere else, not at the active site, okay, that site is called the regulatory site. Regulatory or the allosteric. This button on here. Yes, I know. This site would be called the allosteric or regulatory site. If we're binding at the active site with our inhibitor, then that is called competitive. I'm probably not spelling it right. This is probably an I don't know. Anyways, but yes, all right. That's gonna, we talked about it at least. Okay. All right, I'm gonna clear my drawings. We good? Because you know we're getting ready to get into glycolysis and stuff. All right. Question 8. Aerobic respiration requires what? I hope that this is obvious. I'm going to walk you through aerobic respiration briefly once we get this question answered. Aerobic respiration requires oxygen, oxygen, aero. Cellular respiration is a more generic term. So we can have cellular respiration.
Anaerobic or aerobic.
Exactly. It can be anaerobic or aerobic. And so we have basically the three types of respiration. We have aerobic, anaerobic, and fermentation. Fermentation is also an anaerobic process, but it's slightly different than true anaerobic respiration. Aerobic respiration has glycolysis, Krebs, and the electron transport chain. In the electron transport chain, we use oxygen as the final electron acceptor. In anaerobic fermentation, we have glycolysis, we have Krebs, we have an electron transport chain, and the difference here is we use not oxygen. Yeah, it's usually nitrate, nitrite, sulfate. There's just a lot of other things that we can use, okay? And we'll talk about some of them whenever we get into the lab. The second 8 weeks, we will talk about some of that stuff, okay? But just not oxygen, okay? In fermentation, we have glycolysis, and that's it. All right, we tracking a little bit? Okay, so bacteria that can go through fermentation, or yeast that can go through fermentation, or your own dang muscle cell that can go through fermentation. If there's not enough oxygen there to let the electron transport chain happen, which is the dictating thing here, if we don't have enough oxygen there, we'll just do glycolysis and make some ATP because some is better than none. So generally speaking, net ATP for glycolysis is 2, net ATP per glucose molecule for Krebs is 2, and then the electron transport chain net is going to be about 34. About. Now it depends if you are eukaryote or bacteria, because eukaryotes have to use some ATP to get stuff to the electron transport chain, which is in the mitochondria. So they lose 2 ATP because of that. We lose them because we have mitochondria. All right, are we tracking this? Okay, because you know what's coming next, all right? Which is to say, we're gonna break down the steps. We're gonna start with glycolysis. In glycolysis, we are taking a 6-carbon glucose molecule and turning it into 2 3-carbon. Don't finish the sentence for me. I'm going to call on somebody. John, what is it? The end product of glycolysis. Pyruvate. Which in our body, pyruvate or pyruvic acid, interchangeable term. Probably learn this pretty quickly when you take like physiology and stuff like that. Citrate and citric acid, interchangeable terms. It's just whether or not we are having hydrogens bound onto them and crap like that. I don't care enough about it to make the distinction. So ates and acids are basically the same. Okay, so pyruvate. So we're making 2 pyruvate. During this process, what else do we make? A little bit of NAD, NADH. I want to do a little circle around that guy because he represents something. We're going to talk about that later. And we're also going to make a little bit of ATP. I really wish that I had drawn this differently. Yes, beautiful. All right, we following? That's what I need you to know about glycolysis. You're welcome. No glucose 6-phosphate or anything like that, okay? This is what I need you to know about glycolysis. And y'all still aren't gonna get it. I mean, I love you, but, but yes, this is it, all right? Are we tracking, please? What was our goal? What was the main goal? Like John said to us, what are we trying to make here? Pyruvate. This is our goal. Literally, that's the goal of glycolysis. Now, if you're a fermenter, your goal is ATP. But if you're going through aerobic respiration, pyruvate is your goal. Now, there's a problem here. With glycolysis, you are a fermenter, and this is all you're going to do. You made NADH, and what's that blue represent that I circled there? Electron. You can't just collect electrons as NADH and just build them up and build them up and build them up, and they go to nothing. You have to get rid of them somehow. And so what fermenters do, if you are a fermenter, you will feed that electron back in. Dang it. Back into here. I'm gonna put ferm. Okay, you know what that means. Fermentation. Fermentation. Only in fermentation do we feed that electron back to pyruvate. I don't need you to know all the chemical reactions about what's happening behind that. What you do need to know is when we feed that electron back into pyruvate, we're feeding it in as an H, or there might even be water breakdown or something like that. And that chemically changes pyruvate. If you're a fermenter and you're feeding that electron into pyruvate, then fermenters will turn pyruvate into things like lactic acid, ethanol, and CO2.
And that means it's empty ball.
Kind of, but we're not really focusing on that being amphibolic. So what we could say, if we're talking about amphibolic, then what you could say is you have to go through all of aerobic respiration, even glycolysis, Krebs, and electron transport chain to make ATP. And now the ATP can be used to drive DNA polymerase to make replication of DNA. Because that doesn't come without energy being added in. That needs ATP too. Make sense? So that would be amphibolic. All right. So red is the fermentation. That's it for the fermenters. That's all they do is that. Okay. Next, intermediate step. There's kind of 3 big steps. I still count the intermediate step. You need to know it because it's important for what's happening. In the intermediate step, we are taking our 2 3-carbon pyruvates and turning them into 2-carbon acetyl-CoA. Acetyl coenzyme A. All right, fancy. We're going to make a little bit of CO2. We're not making any ATP, but you do make NADH. Beautiful. That's that step there. And here, just like Lauren said, when I asked where are we making carbon dioxide, and she said we will be making carbon dioxide in the intermediate step and in the Krebs. We don't make carbon dioxide in glycolysis unless you're fermenting, and only if you're fermenting. Okay? So our first carbon dioxide is made here. All right, next step, Krebs. You are so welcome for me not having you guys memorize the steps of these. Okay, Krebs, because I feel like you guys miss the big picture. Krebs, now all we're doing is we're taking acetyl-CoA and putting it in with this big old cycle. Yes, there's a bunch of steps in here, and I'm aware. We're going to bind with oxalic acid, and we're going to make citrate and blah, 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 blah. And that's why it's also called the citric acid cycle. Okay, nice. Cool. But you know what I really care about you knowing? This acetyl-CoA is going to join in with this cycle, and we're going to spit off some CO2 in a couple of places. All right, we're going to lose some CO2s. This is going to make a 6-carbon thing here. And eventually spit off a couple CO2, so we'll be left with a 4-carbon thing. There's a bunch of steps in there that get to this, right? But that's why we know we only lose 2 CO2s in there. And we're also going to spit off some ATP. So there's some products there. And the most important things that we are spitting off here, by far, the goal of the Krebs cycle, we're going to make NADH or FADH2. These are the real goal here, okay? So our goal for the intermediate was to make acetyl-CoA. Our goal for the Krebs is to make NADH and FADH2. And what are those really? Electron carriers. These are electron carriers. Well, what do we really need from them? The electrons themselves. So we need electrons. So what's the goal of the Krebs cycle? To collect electrons. We just happen to make a little ATP and CO2 also. Are we tracking? You're welcome. And don't you wish you learned this stuff like this before you had to learn all those stupid steps in between? Because I feel like this take-home message is way more important than like, it's oxaloacetate, and then it matches with this. And like, I'm just like, cool, but like what's actually happening? This is what's actually happening. This is the take-home message, okay? What's the point of glycolysis? Make pyruvate. Then we turn it into acetyl-CoA. The point of Krebs is to turn acetyl-CoA into electrons. And then of course, what comes after that? The electron transport chain. Okay, so now we're going to erase our big picture over here on the left. Now that we get our point of aerobic, anaerobic, and fermentation, they're all doing this. Well, we know where fermentation stops, right? But the other two are doing this. Now we're going to do our electron transport chain. I wish somebody would have explained this to me too, because when I took biochemistry, it was just all the breakdown and all the steps. And I'm just like, I don't understand what's happening. It's just too much all at once. Let's focus on the major points here. Okay, so glycolysis, glucose to pyruvate. We didn't even lose any carbons. We just took a 6 carbon and split it up into 2 3 carbons. That's it. Then we turned it into acetyl-CoA. Then we fed acetyl-CoA into Krebs to get these electrons. That's where we are. Literally, we just went through all glycolysis and Krebs just like that. All right, where are we at? We've got 8 minutes. Then the electron transport chain. Electron transport chain, in the book it calls it the electron transport system. I have learned it my whole life as the electron transport chain. All right, so here's your electron transport chain. You have a membrane. You have these guys here, some proteins. That's nice, right? And then you have another protein, which is real fancy. It's probably the whole point of everything, so we'll make him green. This obviously is ATP synthase. Okay. All right, so there's our membrane. And if you are in bacteria, anybody know where this is in the bacteria? The cytoplasmic or inner membrane. It's the same thing. Plasma membrane, inner membrane, cytoplasmic membrane, all the same. That's where it is in bacteria. In eukaryotes, this is in the inner membrane of the mitochondria. This is known as the mitochondrial cristae. So if your mitochondria looks like this, it's got all these little brain like wobbles there, that inner membrane is the cristae. Okay, so that's happening inside of your mitochondria. Okay. All right. We said we were bringing in electrons, right? From NADH and FADH2. These guys give off their electrons, but then squirrel their little way through all of this. These orange guys here are called cytochromes. And as these electrons go through the cytochrome, it's a negatively charged electron. And these are proteins. And so what do proteins, how are they going to react? They're going to change shape. They're going to do things. They're going to react to those electrons moving through them. So when they do that, NADH gave off electrons, but it also gave off, I always do purple for it. It also gave off an H+. That H+ was with an electron. It was just an H. Now we've got an H+ because it lost the electron. The electron's moving through, and the H+, as the electron, negative electron moves through, the positive H+ move out. These are all H+. So we're building up all these out here. These move out through here. So this whole buildup of H pluses out here is called the proton motive force. And what does diffusion say? Diffusion, like in physics, we already talked about diffusion says that those H pluses are going to want to come back into the cell to even things out on both sides of the membrane. And they do, but there's only one way back in. Through ATP synthase. Now we've got H pluses. What do we say about H pluses and protein? They change their shape. So now this ATP synthase is going to change its shape and it's going to turn ADP and take some inorganic phosphate and turn it into ATP, which is the goal of all of this, right? This is the goal of the electron transport chain. Build up protons by moving electrons through. Move the electrons through the cytochromes, causing protons to pump out, build up the proton motive force, and then cause the protons to want to move back in, and we make ATP. We got to keep the protons more on the outside though, so that they keep wanting to come back in. So how do we get rid of the protons that build up and they come back in? So this is the whole idea of everybody at least following this up to this point. Okay, so now the H pluses are coming back in. And we've got electrons moving down this chain. They're not just going to, if they move down those cytochromes, they need to go somewhere. Otherwise, they'll just stay right there. So we take the electron plus an H plus, the electron, and an H plus and an H plus, and turn them back into just H's with half of an O2. Well, what does that start to look like? I hope that looks like H2O to you. Half of an O2 is an O, right? And an H with an electron onto it now can covalently bond and share that electron with oxygen to make water. This is why you pee even if you don't drink water, because your body needs to keep making ATP. And the waste product of ATP synthesis is water. So you will pee even if you're not drinking in water, because your body has to make ATP just to stay alive. At some point, it won't be able to do this. It'll break down all the fat and all the protein in your body and add them in as intermediates in any of these steps in here. And then hopefully get as much ATP as it can out of that. But at some point, your body won't be able to squeeze out any ATP out of any of the molecules of your body, and you'll starve to death. Right? So that's a much slower process. But you take away the oxygen, right there, this oxygen right here, and in minutes you'll die. Right? That's why you die when I suffocate you, because you can't make ATP. And now you can see here, the carbon dioxides that you exhale came from glucose. The oxygen in the carbon dioxides that you exhale never touched the oxygen that you inhaled. Are we following this okay? Any questions about any of this? So this is my walkthrough of how ATP is made in the cell. This is aerobic respiration, yes? Okay. All right. We're going to finish I think there's like 2 questions left. We're going to finish them. The gas produced by yeast in alcoholic fermentation. I mean, I circled it on accident. We're going to erase that. Pretend like that never happened. If we're fermenting, we're back up in the red in the top with glycolysis. What's the only gas up there? CO2. That's why your beer has bubbles in it. All right. Question 10, during photosynthesis, plants use what gas to produce glucose as their food? We're going to finish the activity too. Again, this lab today is going to be pretty easy, straightforward, fast. So we're going to feed a little bit into lab time. Just a little. During photosynthesis, plants use what gas to produce glucose as their food? I mean, yeah. CO2. Okay. So briefly, if we're talking about plants, Y'all gotta love a plant, okay? Because you never knew probably, you never thought about a plant this way. But what they do is they basically have a membrane and they have pigments on their membrane, but those pigments are chlorophyll in the chloroplast, and light hits it from the sun, right? And that excites that, and it excites it down the way. That causes H pluses to move through, and this should look familiar now suddenly. Tons of pluses, tons of pluses. Where do the pluses come back in through? It's the same thing. It's ATP synthase. We're going to make ATP a little bit. That's how photosynthesis works. It's not very effective. It only makes a little bit of ATP. But we can take this ATP, and this cycle right here is the light dependent cycle. And we will take this ATP and use it to take CO2 plus that ATP, and then basically turn it into a 6-carbon glucose. What do you think plants do with that glucose? Y'all, you do know plants have mitochondria, yes? They put it through aerobic respiration. Plants go through aerobic respiration too. They just make excess oxygen as a product here, comparatively speaking, but they need oxygen too. Okay, so plants have mitochondria. All eukaryotes have mitochondria, and mitochondria, the only thing that happens in mitochondria is aerobic respiration. No other kind of respiration happens in the mitochondria. The only thing the mitochondria does is aerobic respiration. Are we tracking? It's the electron transport chain specifically. So the aerobic part of aerobic respiration, the oxygen needing part of the electron transport chain. Glycolysis and Krebs can happen in the cytoplasm. The electron transport chain happens on a membrane. Your bacteria, it's the inner membrane of the bacteria. If you're us, then it's the inner membrane of your mitochondria, known as the cristae. Are we following? Okay, any questions about any of this? We're gonna do our activity really quick. You guys did so good with this. I'm so proud of you. All this crap, I'm gonna leave it drawn up on the board really quick for our activity so we have something to refer to. Good job, guys. Beautiful. All right, so briefly, Chapter 10, DNA polymerase is an enzyme that requires metallic ion magnesium in order to function in its role in replication. Sorry that this is so covered in stuff here. What term would you use to label the functional role of the magnesium atom fills for this protein's function? Okay, so if we are DNA polymerase, we are an enzyme that looks like this. And it's going to act on things, but it needs something in order to work. So DNA polymerase needs a cofactor. If it's a metallic ion, we just call it a cofactor. If it needed something like NADH or a vitamin, those are organic molecules. So then that would make it a coenzyme. So DNA polymerase is a conjugated enzyme. Not all enzymes need a cofactor. The enzymes that don't need cofactors, we call them simple enzymes. So there's enzymes, and you are either a simple enzyme or a conjugated enzyme. If you are a conjugated enzyme, you need a cofactor in order for you to work. The whole functional unit of that enzyme plus its cofactor is known as the holoenzyme. The protein portion is the apoenzyme. The non-protein portion is the cofactor. If your non-protein portion, your cofactor, is an organic molecule, then it is a coenzyme. Are we tracking that? Okay. I know, because that got a lot of people in the past. All right, in the Krebs cycle, I know what time it is. In the Krebs cycle, acetyl-CoA, which is 2 carbons, is combined with oxaloacetate, which is 4 carbons, to make a 6-carbon citric acid or citrate. This cycle, which is why it's called the citric acid cycle also. The cycle then proceeds to transfer electrons from its intermediates to make NA to NAD to make NADH or FADH2. Are NADH and FADH2 reduced or oxidized? If FAD is becoming FADH2 or NAD is becoming NADH, what? They're reduced. Gain electrons reduced. Leo says GER. That's the one I remember, and that's what I talked about in the lecture, okay? Now, you need to know it from my class too. I know you learned it in chemistry, but it ain't going away. It literally drives all of aerobic respiration is moving electrons around. So NAD became NADH because it came with an H plus with an electron. So it gained an electron. And Leo says GER, lose an electron and you are oxidized. Gain an electron and you are reduced. The other one was oil rig. Oxidation is loss, and then rig is reduction is gain. Whichever one I learned, Leo says GER, but if oil rig was what you use, then please don't let me stop you. Explain why it is critical to maintain more protons, H pluses, outside of the membrane and not let them build up inside of the membrane. All right, so this is because if H pluses are equal on both sides of the membrane, they don't move through ATP synthase and we don't make ATP, quite literally. So we have to get rid of the H pluses. And we have to get rid of the electrons too that are moving through our cytochromes up here.
But we still need some to keep the gradient.
Yeah, we got to keep the H pluses outside. We can't let them build up inside. And so we need to get rid of them. We have to keep the electrons moving through, but then we need to get rid of them as soon as they move through. So we can just keep the train going. We need to get trucking all day long. The literal reason why you keep breathing and moving and living and growing and doing things is because that is maintained in your mitochondria. As soon as it's not maintained, we start running out of ATP and then we die. So we got to keep that going. And it's fast, it's in minutes, right? So the way that we get rid of those built up electrons and protons that are coming back in afterwards is with oxygen. We split oxygen. It is the only reason why you breathe in air. The only reason. Pretty cool. I think it's pretty wild. All right. Compare and contrast the locations of the electron transport chains of eukaryotes and bacteria. We've already done that. But remember, the electron transport chains in bacteria, it's just on their inner membrane, their cytoplasmic membrane. Our electron transport chains are in the inner membrane of mitochondria, which is essentially the inner membrane of a bacteria that lives inside of our cells, essentially. Okay? So it's the same place, it's just different organisms. Does fermentation generate more, less, or an equal amount of ATP compared to aerobic respiration? Considerably less. So if fermentation is just the red area up here, we only make 2 ATP net from each glucose molecule. Imagine that you are yeast and you are trying to ferment something for you to make you your beer, your wine, or your bread or something like that. And we put you in a situation where now you're fermenting. And all you can do is take your 6-carbon glucose and turn it into pyruvate and make 2 ATP. And then we got to just get rid of that pyruvate and turn it into lactic acid, ethanol, or carbon dioxide, or some variation of those 3. I know you're dealing with that, and that's all you can do, and to make 2 ATP from every glucose. When you have the ability, you have the machinery to do all of this, all you need is oxygen. If we could just give you enough oxygen, and that's why we don't, because we want them to make alcohol and things like that for us. So when they get oxygen, they'll go through aerobic respiration. Fermenters will go through it if they have oxygen. When they don't have oxygen, then they ferment and they only get 2 ATP as opposed to 38, which is quite a big difference. Okay. Anybody have any questions about this stuff? Now is the time. And of course, when you're studying, if you can't figure it out, then email me, okay? So thank you guys for hanging out here. We'll go up to the lab, get settled up there, and today's lab should be a pretty short day. I'll give you guys 10 minutes, so be up there at 11:10, okay?